This week, I had the opportunity to connect with Dr. Christian van der Bos, who is the founder and owner of Mars Ltd, a consulting company in Germany helping companies in commercializing advanced therapies. Christian is a veteran and specialist in MSCs with a track record of innovation in addressing bottlenecks to bring MSC therapeutics into commercialization. In this interview, we hear some of the lessons learned and ongoing challenges that we still face in the field of MSC cell therapy from his perspective.
Hello, Christian. It's great to have you with us today. I'm looking forward to chatting with you about MSCs and cell therapy. I know you have had a very long career in the field of regenerative medicine, starting from your early years with Osiris Therapeutics, with MSCs, to other adult stem cells, tissue engineering, and cell therapy products before starting your own consulting company, Mars Ltd. So tell us a little bit more about Mars and what are some of the type of projects that the company has been involved in since its inception.
Sure. Yeah, great to talk to you, Maya, and happy to connect through these channels. Yeah, as you mentioned, I've been in the field a long time, and I feel like personally I'm at that point where you cease to be proud of having that much experience, and where you start interpreting as being long in the tooth of sorts. So, yeah, as you mentioned, I'm running my own company. It's largely a consulting company. We do some development in the field, and it sort of sprang out of a rather mundane need. I was working with a US company, and we wanted to do clinical trials, and as such, you need a representative in the EU, and that's originally when I founded Mars. And then over the time, I decided to do this full time, and it has been my vehicle of choice since, to provide the consulting services that you mentioned, as well as to finance certain developments that we hope will be useful for the cellular therapy industry. Yeah, we provide services largely in the field of development, of production and quality, and some regulatory aspects too. A lot of our work is in the EU, probably, I'd say 80% or so. Did a substantial stint in India, supporting production at a company called therapeutics, also an MSC company. And some work in the US, and I guess these days I probably have to single out also Britain as not being a future member of the EU. Yeah. So one of the quality-related services that we provide is that of a, I guess, qualified person, as it is called over here, which is the individual personally responsible for maintaining the quality of released drugs. Another project that might be of interest to know about is I had the pleasure of supporting the development and the setting up of a production line for an Ebola vaccine for Merck Sharp and Dohme here in Germany. And you know, it's one of those projects where you don't really have to worry too much about why it is important. You know, so I totally appreciated being able to help there.
Yeah, that's really wonderful. I think, you know, having your own company, consulting company, you get to be involved in a variety of different projects. And I think that's a really exciting aspect of what you do in a day-to-day basis. So that's really fantastic. So, you know, as we mentioned, you have had, you know, done various projects involving MSCs, and, you know, RoosterBio as a company who is solely focused on MSCs. You know, we love to hear your kind of perspective of how do you think MSCs are different from other cell types when it comes to GMP manufacturing. For example, you know, what particular challenges would one expect to face and need to kind of think about before they go into GMP manufacturing? So if you could share some of your experiences, we'd love to hear it.
Certainly. Well, if we perhaps start with another example that might help to sort of clarify matters a little bit. If you, for example, consider hematopoietic stem cells, that's a set of cells that happen to be defined by one or two or three very well-defined markers. And we also happen to have very distinct expectations of these cells. So we would like to infuse them, and they are supposed to set up a new blood system. And we can follow all of that with, a army of CD markers, and that's a process that we have pretty much under control. For MSCs, in contrast to that, I would say, we don't have the luxury really of having a particular defined markers. Most of the people in the field still work with the Dominici panel. And what is perhaps making matters more difficult is that our expectations are also quite diffuse of these cells. There's not a single thing that is being asked of these cells, and so as a consequence, providing proof that they are living up to these expectations is also, let's say, complex. I think, the MSCs were perhaps outside of the hematological field the first major push in regard to cellular therapeutics. And as such, many of the pitfalls in doing so were discovered with MSCs. There was also, I think, at the time, sort of a certain mystery shrouding anything that was referred to as a stem cell. And that, in my assessment, proved to be yet another pitfall for MSCs. So in that context, early descriptions were referring to their ability to find diseased tissue, to become that tissue, to replace disease, destroyed, and so forth tissue, and no matter exactly what it was. And granted, there was some initial evidence for that, but I think in retrospect, it's probably true to say that we overburdened these cells as a new pharmaceutical completely with expectations that were sky high and consequently were not met in every case afterwards. And so I think there was a distinct sense that they were living not up to expectations, but that was sort of built in because the expectations were just A, much too diffuse, and secondly, way too high. So I think now after these, what is it now, about 25 years since their sort of rediscovery, if you will, we have a much more clearer picture. We know that they're good for particular applications. We know that they can make a difference in revascularization. We know the same to be true for graft versus host disease and similar effects. And we know that they produce a massive quantity of bioactive factors and of extracellular matrix. And so pretty much any setup that combines these properties or the benefits inherent in these properties is likely to benefit. And so I'd single out those two areas plus orthopedics as perhaps the most promising applications. And so with that in mind, I think we then are only a step away from coming up with quantitative descriptions of these cells and measuring their true benefit. But the markers that I mentioned earlier are useful to segregate these cells, for example, from other components of their source tissue, be it fat or bone marrow. But in my view, they don't really have a lot of diagnostic value. So, whether some of these markers are expressed at a higher level or at a lower level, at least as far as I know, does not really correlate with whether these cells are particularly well suited for this purpose or for the other purpose. So, and once you have singled out a particular purpose, then it becomes a lot easier to come up with something useful and quantitative. For example, in the field of dampening immune responses, such as in GVHD, the enzymatic activity of an enzyme that catabolizes tryptophan, for instance, IDO, has shown itself to be a really useful measure, and it's a nice and quantitative assay. So that's, I think, a significant step in the right direction. It's probably not going to be the only thing you want to do in that scenario, but it is much better, much more quantitative than, for example, some sort of differentiation assay or so. still
absolutely.
found in the area. And if I may add one more sentence there, without stretching it too far, these features, that is, making either extracellular matrix or secreting certain bioactive factors and so on, in my perception, are largely dissociated from any kind of stem cell aspect. Right? It's just an inherent property of these cells that they make, for example, an enormous quantity of VEGF, and that is helpful for making new blood vessels. Whether these cells have the ability to differentiate into something else, I think that is almost beside the point in this case. And I'm stressing that because in descriptions of these cells in the context of developing pharmaceuticals, these differentiation tests still pop up again and again. And them being complicated, cumbersome, lengthy, and not quantitative,
right.
I would strongly recommend to ignore them. Right.
Yeah. I
that
think
was a rather long-winded answer to your question, wasn't it?
No, absolutely not. I think it's great to hear kind of your perspective on different aspects of it. I think we have had definitely many discussions around potency assays, right, for MSCs, because
Yes.
as you said, they have such a diverse kind of profile or portfolio. It really depends on the therapeutic indication that you kind of targeting. So I think that is really critical in identifying what are you using it for, really, at the end of the day. So so having seen sort of the field evolve over the years, where do you see, what are the lessons that you have learned, and what do you think the future will bring for us, especially with sort of the evolution of MSCs and recent developments with COVID? Do you think this is going to be a positive thing for MSCs to come back in fashion, so to speak? What are your thoughts on that?
So starting with the latter, I think what we have seen now in the development of drugs to treat people with COVID-19 is that anti-inflammatories, well-established drugs, do have a clear benefit, and it's probably within that realm that we can also expect MSCs to provide a benefit. Whether that will totally materialize is yet to be seen, but I think that is a potential utility for these cells that is well based on the theoretical assessment and on what we know of these cells to be capable of. So, I think within a few months we'll probably see the results. There's been, I don't know how many trials started using MSCs for this particular purpose, and so given this sort of shotgun approach, I hope that we will be lucky and see a significant benefit here. So just to be clear, I have no expectation that these cells would prevent you from falling ill with COVID or contracting the disease, but I would expect that there's a significant chance that they help you cope with it better, similar to those anti-inflammatory drugs that we mentioned.
Yep. Awesome. So you've been in, well, I guess you're very familiar with the policies in Europe. I'd love to hear sort of what do you think is the view of folks, researchers, industry, academics, of MSCs as an advanced therapeutics? Do they see MSCs potentially as a promising sort of chassis for advanced cell and gene therapies of the future? What do you think is their perspective?
I would probably put out a strong yes to that question.
That's great.
These cells have been shown to be of a really excellent safety profile in thousands and thousands of patients, and they have been shown to produce all these bioactive factors. They've been shown to produce large quantities of vesicles, which have attracted a lot of attention over the last, say, 5 years or so. And so between them known to be safe in various applications and having these attractive features, I think they are clearly in the field of interest. Now, I suppose one reason why you might ask that question is they have been around for quite some time. When you go back to the early works of Friedenstein and colleagues, that was in the early 70s, and then came that sort of rediscovery by Arne Kaplan in the mid-90s or early 90s, and so early clinical studies, I guess, happened in the mid-90s too. So we're looking at basically 25 years of development time without a really major breakthrough. Compare that to, for example, CAR T cells or so, right? But I think you have to put that into perspective for the reasons that I mentioned probably in our first segment, where probably every mistake that you were able to make in developing a cellular therapy happened with these cells because simply they were the first real serious stab at it. So I look around at clinical trials and at company profiles at their current developments, and I feel there's a lot of really sensible stuff going on these days. I rarely see things where I would think that there's no logical basis for applying MSCs to a particular situation. So that gives me a lot of optimism about the next few years and the clinical results that we will see. I think what we need to keep in mind is that we are talking about live cellular APIs here, so active pharmaceutical ingredients, and that, I mean, as banal as it is, oftentimes when it comes then to actually putting them to use in the clinic, that is again overlooked. They are being passed through small needles, this, that, and the other thing happens, all of which has the potential to damage these cells and reduce their potency. So it's, I think, another lesson that we can take away here is that the developer of a pharmaceutical based on MSCs needs to cover the production and application from A to Z, and they need to be there in my assessment also when the material or when the final therapeutic is applied. And that needs to be part essentially of the production process so that you have full control over how these comparatively fragile APIs are treated until they are well within the patient.
Right.
Yeah. There's a couple of other things. We take for granted that we can cryopreserve these cells, and depending on in which context you do it, some in research labs you might even be as straightforward as putting them into a styrofoam box, then slowly freezing them down on -80, and when you thaw them up, you will see that a lot of cells are still viable, and then the conclusion, without being spelled out fully, is that basically you can do whatever and these cells will be alive. But between maintaining, say, membrane integrity and full functionality, there's quite a bit of space. And I would put out that cryopreserving these cells and just looking for viability after such a step is probably also another hurdle you can put up in front of the potency of these cells. So none of these things is particularly complicated, but I think in the routine processes that are sometimes geared towards output, towards not spending too much time with the patient and so forth, it is easy to overlook some of these aspects. And then there's, I think, a significant danger still out there that if we do not address these points properly, we end up with clinical trials of mixed results.
Right. Yeah, that's a very good point, Christian. I think I definitely agree with your assessment and perspective on those points. So thank you for sharing that. I guess having worked in both like EU and the US in the field of regenerative medicine, do you think the regulations around advanced therapies are easier or more difficult in the EU compared to the US?
Well, one very obvious difference is that in the EU, everything that goes into people needs to be full GMP, right? And with the FDA, in my experience, that's a matter of negotiation and of how advanced you are in your clinical study program. The latter does sound like an easier path, but I've been witness to lengthy such negotiations where you go back and forth and back and forth. And so I think you can probably live in both fields. What I appreciate about the US situation is, for example, that the FDA is reasonably aggressive in regard to snake oil folks who put out a shingle saying stem cells and then do whatever. I'm happy to report that that has caught up here too, and I think we have seen a drastic reduction of ill-fated attempts to make use of the label stem cells for unproven purposes. So I think that works in both geographies. I guess I'm a little biased to the EU setting because simply I'm spending most of my time in this setting, and hence I'm quite familiar with it.
Right, absolutely. That's great to hear. Any last advice you would like to give someone who's trying to develop cell therapy, perhaps some pitfalls to look out for?
Yeah, so I'm going back to our first points of discussion. I think if you would like to avoid disappointments, stay quantitative, stay away from a bowl, and really rely on your facts. Pretty much anything where you make an assumption is going to come back and bite you somewhere.
Good advice. Great. Thank you so much, Christian. It's been great to connect with you today and chatting with you about MSCs and cell therapy. So really appreciate your time.
Thank you, Maya. Talk to you soon.
Talk to you soon. Take care.